Professor Peter Doherty. I appreciate your time as always, Professor. Thank you so much. Last time we spoke, you were optimistic about the prospects of a vaccine. Do you still feel that optimism?
Yes, I'm optimistic with reservations, of course, because this is a new virus and we don't know that much about it. There could be some traps there for us that we think might be there, but so far there's no evidence they are from anything we've done with this virus. And I would also echo what the Minister has just said and say that we are fortunate in that we have the science to develop novel vaccines, which we're doing, and that we have, due to the fact that CSL is an Australian-based company and has shown great loyalty to this country, we are well placed to be able to produce vaccine also. I was concerned about that actually until I talked with Andrew Cuthbertson from CSL, who told me that that is indeed the case.
Well, that is good news. The Chief Medical Officer, Dr. Murphy, has been saying that the likely time frame is 12 to 18 months, but given how many experts, how many of the best in your field researching this are looking at it around the world on this one initiative, that it could be achieved sooner. What's your sense on that in terms of the timeline?
Well, if all goes well, there is a British vaccine that comes out of the Jenner Institute in Oxford. It's what we call a virus-vectored vaccine, which is a fairly established technology that's been injected into humans, though not, of course, with the SARS-CoV-2 spike protein in it. It's called a chimp adenovirus vectored system. Now, that has been tested in ferrets at our what was the R laboratory in Geelong, ACDP, I think it is now, and then it's been tested in monkeys, in macaques, and it's already gone in, I believe, to about 600 human volunteers in England, where, of course, they are exposed to the SARS-CoV-2 virus because it's circulating there. So we're well into what you would call a phase 1 clinical trial in humans. Now, the only way, once you've started to put a virus into humans, the only way you can really test it is to put it into more and more people, so long as it seems to be safe and seems to be protecting people, to see if that holds up. And the British have already organised, I believe, to get 60 million doses of this vaccine made, and it could be going into people's arms in substantial numbers as early as September. Now, of course, that all depends on it working and being safe.
Yeah, well, that was what I was going to ask you about, that safe component, because from what I've read of what you've said in recent weeks is that the problem with this COVID variety, unlike SARS, it doesn't grow nearly as well in monkeys. Therefore, the test system is not as accessible.
It's not as robust in that sense because we would like to have a really big virus challenge. So it doesn't challenge quite as well as SARS does, or the MERS virus, the one that came out of the Middle East. But and some of the vaccines that were made along the way as experimentally for those viruses, when they were put into monkeys, were causing some bad consequences. Not all of them, just some of them. But we haven't seen any of that yet with this virus. And really, the ultimate test is to put it into human beings. I know there's a risk there. I mean, that's part of life, really. There's a saying from the old HIV science that mice lie and monkeys don't always tell the truth. And the only truth of what happens with human beings is, in the end analysis, what occurs when this vaccine confronts the virus in a human being.
Yeah, indeed. And I guess, particularly given what a calamity the UK have faced, that they'd be more willing to take some risks. With the HIV response, it was done with antiviral drugs. Do you see a scenario where that might be the course of action for this, or would that run parallel to, say, a vaccine as well?
Absolutely. I mean, our own institute is involved in at least 3 vaccine candidate developments. But the other thing we're doing is testing a large number of potential drugs. These are based on existing compounds and also designer drugs that are being made by people who actually, as with the AIDS drugs, actually design a drug to do the job. And they do that by what we used to call X-ray crystallography, we now call structural biology. This is a technology that uses the synchrotron at Monash University that we built some years back. We're well placed because we have the synchrotron and so forth. And so we're testing a lot of these drugs at the moment in what we call in vitro systems, that is growing viruses in cells. We're just discussing how to expand that and how to make sure we've got the animals available to do the preclinical testing, which is mandated for all drugs and vaccines by regulatory authorities like the American FDA and our TGA. Now, that we're sort of pushing forward and making sure we've got the necessary infrastructure. But the point is that if, for instance, the vaccine didn't work, and it never worked for HIV, we don't have an AIDS vaccine for reasons due to the complexity of that virus, which we don't see with this particular virus. But it's possible we won't get a vaccine. And it's also possible if we do get a vaccine that it won't work very well in the elderly. People of my age don't do necessarily all that well with vaccines. And the way to protect them may be with drugs. So if we can make drugs, and I think we need to push on drugs just as fast as we push with vaccines, and of course that's going on across the planet. If we can make drugs, we can both hopefully treat this infection a bit better, because if we could treat it, we can just treat it like any other disease, and we don't have to do all this elaborate social distancing and stuff. But we're not at that stage yet. Not only that, if we have drugs that we can use to treat the infection, we could use them as preventives. And that's done with AIDS. It's a procedure called HIV PrEP, where someone who's putting them at high risk, themselves at high risk of catching AIDS, can take a drug called Truvada, which has 2 antiviral drugs, anti-HIV drugs. And they can take that every day, and they're not going to catch AIDS. We can do the same thing with older people with COVID-19. And as most older people are taking at least 3 or 4 drugs a day, an extra one wouldn't make much difference.
Yeah, fair point. And Professor Doherty, when you look at this situation at the meatworks in Melbourne, for a layman watching this, should we be worried about a cluster like that, or is this to be expected even more so over coming weeks if we have some of the social restrictions eased?
Yes, it's been an interesting. I'm not privy to the exact details because it's operating at a level above my pay grade. I'm basically involved in the lab and doing communications more than anything else. But it's been an interesting exercise, I think, for testing our contact tracing capacities and making sure we've got all that in place. And so that's what's going to happen. As I expect that's what will happen. As we lift the restrictions on social distancing. Now, it'll be interesting to watch because the virus looks as though it may have almost been stamped out in South Australia and maybe Western Australia, maybe the Territory as well. So it could be when we lift social distancing, we won't see flare-ups. I'll be very surprised if that's the case in the eastern states, so forth. Of course, with Melbourne and Sydney being so much bigger, and being more exposed to international travel, perhaps. So we'll see. But if we get little outbreaks, we get flare-ups like the meatworks, our hope is that we can put out those spot fires, keep the numbers of cases in the hospital within manageable levels, and not overwhelm our medical system and our hospital staff and all those frontline personnel. That's the aim all along. There's never been an intention here to try and eradicate the virus, though we may be luckier than we've thought. But we're going to find out when we lift those social distancing measures.
And just finally, Professor Doherty, as someone who's dedicated your life to dealing with these sorts of problems, viruses and diseases of this nature, are you pleasantly surprised at how well Australia and, for that matter, New Zealand have done in dealing with this pandemic, one which we haven't seen in our lifetimes?
Yeah, yes, I'm not only pleasantly surprised, I'm a bit delighted because I think we've been a bit lucky. Australia, the lucky country. Um, I think, though, what happened is we didn't get this virus established here in the community so early. What we now know is that the virus infected someone in France in December. Now, that's much earlier than we thought. And the French didn't react for at least 6 or 8 weeks after that. So that virus was spreading in Europe for a very considerable time before the authorities reacted. It was spreading in the United States, we know, for at least 6 weeks before they reacted in any sort of comprehensive way. But we got onto it early here due to the fact that Chinese gave out the gene sequence of the virus early in January. That was all we needed, actually, to start dealing with this thing, but it allowed Mike Catton at our institute and others in Australia to develop the PCR test, the test that we use for detecting the virus. And when we got our first case in 25 January, it was, the person who had the disease had just come back from China. They were very aware of it. They immediately called Monash Medical Centre, I think it was, said, I think I may have this infection. They were brought in under secure conditions, tested, and it was found to be so. So that was our first case. That was the person we isolated the virus from. And we were actually the first group in the world to give out that virus to other labs across the planet. And that's how science works. We collaborate across the planet. And we want to keep that collaboration going. So I don't want to see tensions developing between different countries and that sort of silly game that some people are playing.
So yeah, but just to elaborate on that, you want cooperation at this time, particularly in the face of the pandemic. Would you urge all concerned to ease some of the rhetoric?
I think the rhetoric is very counterproductive. Some of it's coming out of the United States, and it's due to the fact it's an election year and they're beating all sorts of drums, as always happens. I don't think we need to get involved in that. I've been delighted to see that the Prime Minister is making really sensible statements on this. He said very recently that our intelligence agencies have looked into it at the government's request, of course, and they think it's pretty much come out of nature, and that the chance that it sort of was a lab escape is at the most 5%. So basically, that's something we'll need to work out where it actually did come from. But quite frankly, as we try to deal with this crisis and we try to get us out of this situation, both economically and medically, it really doesn't make any difference to know where the virus actually came from. And the last thing we want is people getting into silly blame games about this. It's counterproductive. It doesn't get us anywhere.
Nobel laureate Peter Doherty, Professor Doherty, and your institute in Melbourne, all strength and power to you. We hope that they could come up with a vaccine, and your colleagues around the world do as well.